So welcome everyone to this evening lecture. This is the Fall 2018 Regenerative Medicine lecture, and our speaker tonight is Dr. Kevin Healy. Dr. Healy is currently the Jan Fandrianto and Selfia Halim Distinguished Professor in Engineering at the University of California at Berkeley in the departments of Bioengineering and Materials Science and Engineering. He received a bachelor's degree in chemical engineering from the University of Rochester and his master's and PhD degrees in bioengineering from the University of Pennsylvania. Dr. Healy works at the interface between stem cells and materials science. He develops dynamic engineered systems to explore both fundamental biological phenomena and new applications in translational medicine. Dr. Healy is an elected fellow of the American Institute of Medical and Biological Engineering, the American Association for the Advancement of Science, and the Biomedical Engineering Society. He has authored or co-authored more than 350 published articles, abstracts, and book chapters. He's also a named inventor on numerous issued United States and international patents relating to biomaterials, therapeutics, stem cells, and medical devices. He's founded several companies to develop these systems for applications in biotechnology and regenerative medicine. This evening, Dr. Healy will be presenting Innovations in Tissue Regeneration. Please join me in welcoming Dr. Healy.
Thank you very much. It's quite a lovely introduction, totally unnecessary though. Well, thanks all of you for coming. And I hope I have this at the right level, that you'll enjoy it, you'll find it a little stimulating scientifically, maybe deep scientifically at some point, but nonetheless I hope I engage your interest. I'm required to have financial disclosures. There they are, that's all I have to say about them. Okay. So my lab does a lot of things, and it would be impossible to cover all of that tonight. So we work in these areas we call biointerface science. That's with cells just sitting kind of on a plate, something flat like the desk. I'll talk about this tonight, so I won't say much more. We also make these, what we call tissue models, and I'll talk a little bit about those at the end. And so to get to what I'd like to discuss as a matter of introduction, idea of this review of tissue engineering and sort of how we got to where we are in the field of tissue engineering. And I'll describe then what we call our advanced scientific materials. In this case, these are what we call growth factor sequestering hydrogels for regenerative medicine. It's quite a mouthful, right? But you'll see why we have to make things that are inherently quite complex to get to where we want to go. Hopefully with enough time then, I'll talk about these in vitro human tissue models for regenerative medicine, and that's because this requires a lot of animal experimentation, which we're trying to avoid. So we'd like to basically use these kinds of systems to move forward. Okay, so let's take a step back in time to 1993. Some of you I could tell weren't born. Some of you I could tell were born. Those two guys over there. You know, this is the time I was just finishing grad school, and a scientist named Robert Langer at MIT, another scientist, Jay Vacanti, at Mass General Hospital in Harvard, they came up with this concept of what's called, and it has become a field and really an industry based on what's called tissue engineering. And that is, you take biological cells, you combine them with some sort of material. This is essentially a polymer scaffold. I'll describe what these are in a moment. These are pictures of what these look like. When I use scaffold, think of something porous. Matter of fact, this structure is very similar to those green scrubbies, those 3M scrubby pads. It's kind of porous. You hold up to the light, you can look through it. That's what this material is sort of like. The difference is it degrades in the presence of water. So as the cells are contacting this structure, they're making all sorts of matrix molecules, proteins, for example. And those proteins basically fill in these spaces. The cells fill in these spaces. And you essentially can make a tissue. That was the argument. You can make things like new bone. Early on, the darling was cartilage of engineers and tissue engineers because cartilage is privileged. You don't need a blood supply to it. It receives all its nutrients by diffusion of oxygen and also exchange. So cartilage was fun to make. Liver, you can see all these tissues were supposed to be made, and we're supposed to replace your tissues if you have disease or damage. Tissues damaged maybe through trauma or disease, either genetic or acquired during your lifetime. So you would wonder, 1993, where's all this good stuff, right? And I used to have a cartoon describing pearls and swine, sort of saying, yeah, it's a great idea, but it's really hard to do in practice, right? So what's hard to do? Well, we need host cells that can, I could see this as a mixed audience, so I'm going to have to use terms, and maybe your friends can fill you in on those terms in a little bit, but the cells have to differentiate into tissues we want. Basically, a bone cell has to become a bone cell that produces bone. A hepatocyte, which is a cell in the liver, has to become a cell that produces liver-type tissue. And it has to do it in specific architectures. We need molecules called growth factors, very potent molecules that teach these cells what to do and basically how to process and understand their environment to make the tissues of interest. And then this is where a lot of us, meaning engineers, came along to develop matrices that are supportive of cell growth and attachment and differentiation. And many of these boxes have been checked. The problem that we ran into, you'll see in a moment, is largely how much these things cost. And I'll give you this example. One idea was to make a tissue-engineered blood vessel. This was sort of the holy grail of tissue engineering because one could see you could make a thin little tube, and the idea was to replace basically coronary arteries or post heart attack or infarct, to replace those blood vessels. Don't worry about the chemistry. This is just a polymer that we use. This is much like if anybody has a fleece on, you can, sort of a synthetic fleece, right? I'm sure many of you know what those are. I'm trying to look if somebody has one out there. That's how this is made. It's basically, again, a porous structure. These polymeric materials, they degrade by exposure to water. And so what that means is when you put cells in them, they actually remodel this material. They produce their own natural proteins, and eventually you have a tube that's, instead of synthetic, which is what you would normally use to replace a blood vessel, that the material is typically similar to Teflon, that you might know about, or Gore-Tex. So Laura Nicholson, who came out of the Longer Lab, you can see we've moved to 1999 at this point, made a tubular structure. This is basically just the wall thickness. So this would look like a big tube like this in cross-section, and you're just looking at that little part there. So what she did, let's just look at this little component. This is basically, this little piece here is a small tube that's hooked up into basically this bioreactor. I won't have time to describe it, but you could see that basically fluid flows through these systems. It bathes the cells naturally, and over time, basically, you add pulsatile flow, and the tube expands and contracts, and in that case, then you form this tissue, right? So you would think that's a great idea, but the problems that occurred were complications of expense, and where do we get the cells from? So if I wanted a graft, if I had to take it, the cells, we had to take a biopsy from one of you for the cells, then we have basically an allergenic tissue, if you will, and immunosuppression becomes a big issue, like organ donation. So sourcing of cells became a problem. So what folks thought about was then, okay, here's our structure. We added our cells, we grew this up. Now, what they did is they used a chemical cocktail to get rid of all the cells. So now the cells are gone. So what's left is this protein matrix that the cells have made, and that's a solid tube, and you can use that. And this is it. This is what's actually in the clinic, and the California Institute of Regenerative Medicine has supplemented this work to the tune of $11 million to actually basically support a company to go through the clinical trials of this tube, or this product. So we call this the regression of the tissue-engineered blood vessel, and that's sort of a snapshot of the field of tissue engineering. So we started out thinking we're going to make basically a repeat of the blood vessel. It's costly, it's complex, okay? So what goes this way is simplification and design as we go down this way. So basically, this is trying to get age-matched and risk-matched cells in clinical trials. This proved difficult. And then other things like, well, maybe we'll just take the cells out and add cells along the lumen, the center portion. Those are cells that would be normally the cells that line your blood vessel. Those are called endothelial cells. And even that didn't really take. What has really taken in terms of this system is actually creating threads from those proteins and actually weaving those threads to make a conduit. Okay. So the problems that the field ran into were namely these two main problems, that you have basically oxygen supply to tissue. You can't make a massive piece of tissue outside the body because you don't have basically blood vessels. In your body, every blood vessel, a minimal separation from what we call a capillary vessel, is typically on the order of about the width of a human hair. It's pretty small. So you can imagine if you were saying you're going to make a piece of bone, you're going to make a liver or a kidney or whatever, that's macro scale, right? Inches or centimeters, right? So you can see this is a major fundamental limitation of what we call ex situ tissue engineering. That's the tissue engineering process where we use that sort of bioreactor design to create this what we call tissue equivalent or a product. They also lack nerve instruction, so that's a problem. A lot of times you need this neuromuscular junction, and you need some way in which to activate if you're making muscle, cardiac muscle, skeletal muscle, things of that nature. So what came next? Well, it's called in situ regenerative medicine. And that's designing systems where you can actually involve a material, cells, to actually handle a problem. So let's take a very important one. This is a heart. If you have a blockage, if you eat too much, I don't know, Ben & Jerry's ice cream or something, you put yourself in this scenario. There's very interesting things that go on here I won't have time to tell you about, but this is where your wall thins because of lack of oxygen. And a number of scientists, myself included, said, okay, years ago, we said, let's inject basically hydrogels or stem cells into this environment. Why a hydrogel, I'll explain in a moment. It can have stem cells or not, but the end result has to be that these cells and or the material lead to what we call blood vessel genesis. So you call that angiogenesis, so creation of new blood vessels. And this leads to proliferation and this idea of reduced cell death. This is supposed to be a movie. It was supposed to play. It's a needle. It's going into a heart and injects a thermoresponsive hydrogel. I'll talk about something a little bit different, but this gives you an idea of what we were trying to do, that this infarcted area here would get a material that would stimulate the growth of new blood vessels and actually repair that tissue. Competing technology is, okay, the same idea that I just described that I said didn't work well, which is basically ex situ tissue engineering, and that would be maybe making a patch of the similar type of construct I just said and putting the patch over your heart. Okay. The patch and in situ tissue engineering are competing technologies. I'm banking that this is the technology that will move forward. Okay, flames, right? If you're bored and you see the flames, I tell this to the students, that's the time to pay attention, take notes, okay? So what did we see when we injected this hydrogel? We saw only a modest improvement in cardiac function. This is a big problem, will be the message for the rest of the presentation. The transplanted cells don't survive. With or without a hydrogel, you're maximally looking at a couple of, without a hydrogel or without a material support, you're looking at somewhere between 24 and 72 hours that basically the cells survive and they die. Anybody who read the front page of the New York Times this week probably came across a very famous scientist in this field that was accused of misleading the scientific community with falsification of data for over a 10-year period. And that didn't help with this particular application as well. And then one thing we observed, this is something we'll get back to later on this evening, is sometimes the hydrogel itself, without any cells, was actually going to be more beneficial than just basically the hydrogel with cells. And that might seem counterintuitive, but what you need to know surrounding this environment here, this tissue environment, is a lot of also healthy tissue where it can grow into this region. And we'll talk about that briefly. Okay, so a little bit more now on the scientific part. So that's why we're here, right? That's how we got to this state of tissue engineering that's covering largely almost, if you will, a 25, close to really a 30-year period if you go back to the early conferences in tissue engineering in 1988. Okay, so why do we make these complex hydrogels? I'll describe in a moment, and you'll see, I hope that'll become obvious. So look, a cell sits in, basically, this is a biological cell. It sits in this extracellular matrix space. These are proteins. It uses these, what we call cell surface receptors, these molecular, usually they're heterodimers, so they're two proteins coming together, to bind to those proteins. Some of these signals, these particular signals called integrins, they communicate directly to the cell via mechanical linkages. That's called the cell cytoskeleton. And others, like this material, this protein called the growth factor, does that through some sort of biochemical sequence. You have two then inputs to the cell that are widely studied. One is basically a physical linkage for adhesion, the cell attachment points, and then basically the proteins we call growth factors. And I'll highlight this example here. These growth factors are drawn specifically so they're interacting with what we call proteoglycans, or polysaccharides within the extracellular matrix. So extracellular matrix is outside the cell. Obviously, intracellular is inside the cell. This one here, this is what we call a rogue growth factor. It's not in the right position. It can do a lot of damage by not being coordinated specifically in the matrix properly. And I'll point to some studies we have related to that. So if you use this as a roadmap, and then so information then basically cells need a matrix to survive. That's clear, right? So we spend a lot of time designing this part. So if I take away the cells, what do I have? I have that matrix. In essence, that matrix itself has different properties that I can look at. So cell binding domains would be one of them. That's basically on the protein small amino acid sequences that the cell uses through those integrin receptors to attach to the matrix. So it can kind of cruise around and walk, right? We call that anchorage-dependent movement, okay? We also want to know things like its material properties, like how stiff it is. Like cells might grow on something like this, but if you pull on your skin, you can just tell. You don't have to be a material scientist to realize there's a difference in mechanical properties and stiffness between this lectern and your tissue, right? So we call that modulus, basically, relevant material properties. Those are the symbols we use for what we call elastic and shear modulus. I talked about growth factor presentation, having it what we call solid phase presentation, and then degradation kinetics. And I'll touch on all of these briefly scientifically. So we're going to do a little bit of a deep dive. So put on your air tanks and come down into the sort of molecular chemistry space as we sort of develop these things. So this picture, before we get there, this picture takes the path of basically at the scale of basically, like, I'll use something practical like the width of a human hair and even smaller, you start to see this is a material and this is the cell interacting with it. And you need to design your material to have properties that are biological in nature so that the cell sees the material as some extension of itself, like this biomimetic strategy to make it seem like it's seeing the extracellular matrix. The path we'll take tonight is getting into basically this kind of material, hydrogel. I talked about the early tissue engineering examples with those porous solids. There's other nanofiber type structures. If you are in this field, you might know about these things. I'll explain in a moment why I think hydrogels are quite valuable. And then we already mentioned quickly material mechanics. These things are hydrogels, and some of these other materials can be designed to be much softer than these very hard materials I've been tapping on. So why hydrogels? Well, first of all, you can synthesize these or extract these. We'll talk about both of those as an example. That's what a piece of hydrogel or powder might look like. You add water like this, right? And then if you put them together, they make what is called a hydrogel. It's a physical entity. It's a material. This little cartoon just describes, or I say images, this is its dry state, and as it adds water, it basically swells. So these are on order of 90 to 95% water by volume, depending on how they're made. So they can have a large amount of water, which is very similar to your natural body, your structure of the extracellular matrix. You can do other things. You can put hydrogels, these colored hydrogels, or within a hydrogel. That's kind of what you're seeing here. And here's where we're going. Cells that are stained with a fluorescent probe, they look red. They're in the hydrogel as well. And they like the hydrogel. So what kind of hydrogels are available? Well, you can get seaweed. One's called alginate, chitosan. It can come from excess crab or crustacean shells. We use hyaluronic acid. You can get that from the rooster comb, but it's also now made in bacteria, basically through biological engineering. And so that's the polysaccharide that we use. This is your native tissue, and the idea is, how do we take this to look something like this? So this is what we call collagen basement membrane. It has all these parts, proteins, that I won't go into in detail at the moment, but we'll hit upon that in a moment. Some of the relevant points. So hyaluronic acid, it's a popular material. These are all colleagues around the world that are also working with this material. It has interesting properties. One is that we can start to chemically modify it to handle some of those points that I was trying to emphasize in terms of what a biologically inspired system would look like. So in this case, you have molecules for adhesion of cells. You have cross-linkers that give you the mechanical properties, and you have molecules for growth factor sequestration. So it looks something like this. So for those of you that are not involved in material science at all, these materials, or these parts of the system, is called a cross-linker. Their number, if I draw it like this, maybe more in a cartoon phase, as we link these polymer chains together, these cross-links impart these mechanical properties. And it gives us the stiffness in which we can control something that's relatively soft to really soft to something that's more like a biological tissue. These molecules are for adhesion to the cell surface receptors, integrin receptors, and this molecule is heparin, and the heparin here, I'll pick this one, is there to present the growth factor in a biologically important way. These cross-linkers are not only interesting from the standpoint of actually making the material, they're degraded by cells, and that's a very important part. They're degraded by cells as the cells wish for them to degrade. So this, we call it cellular demand. Remember the first materials I said just degraded by random hydrolysis, exposure to water? These are much more sophisticated materials, right? So what we do is we take that hyaluronic acid, we chemically modify it with these groups, which are basically acrylate groups. We use a reaction called a Michael type addition. This has a SH, that's called a thiol. These two react, so now it's chemically coordinated. We do the same with heparin. So we have what's called a two-part system. We have these components in one part of this syringe. It's called a two-barrel syringe. We have cells and these cross-linkers in the other part. So I should make that maybe red or something. And then this tip mixes them. And so this is mixed, and all those things get mixed together. And so at the time, it's rather a fluid-type system. But then the reaction, this reaction between now these thiol groups and these unreacted species allows for this cross-linking event to occur. And so now the cell is trapped in this basically hydrogel. It's just colored blue because, I don't know, it's from Berkeley, so we made it blue. But this is important technologically. This is the rheological properties. And this is when it goes from a liquid to a gel. It takes about 2 minutes. You have about 5 minutes of working time to inject this into different tissue defects. And then it sets up and it gets stiffer as time goes on. Okay, so let's talk about some of these important properties. Let's start with thinking about this idea of growth factor presentation. So this will get a little technical, but I think it's really important for everybody to understand that. So we make this system. These materials, as I said, I can't draw the water in there, but imagine what's in between all these polymer chains is water. And so we have something we call a mesh size. I'll try to draw something that looks like that. That's what we call the mesh size. The mesh size just means that's like an open water pore in the network, and that allows both drugs and other molecules to diffuse through the system. They can come into the cells, and the cells can produce things that come out of the system. If we do this with a simple control protein, bovine serum albumin, has no interaction with our hydrogel, you see it comes out of the gel very fast, within basically hours. But if we use this idea of using the heparin to conjugate or I should say associatively stabilize this particular growth factor, which is transforming growth factor beta, what we end up doing is having retention times on the order of days, about 20 days. You can see about 70% of the drug, or in this case, the growth factor, is retained on this kind of system. So this is the size, about 3 to 5 nanometers, 4 nanometers, so there's like a 1-to-1 pairing here. And we get this kind of response, which is quite nice. If we take cells, these cells are derived from a cardiosphere, which is a biopsy from the heart that can grow out. You can get these from experimental animals, which is what this is from a mouse, but we've also worked with patients that have had different types of surgery, and we've used their what we call cardio-derived cells. This is just letting you know that these cells can become important cell types. They can become the heart cells, cardiomyocytes. They can become endothelial cells and also smooth muscle cells. These cells, what we call CD31 positive cells, those are endothelial cells. Those are the cells that are going to be the trick for us to actually get stem cells to survive when we transplant them. So we're going to exploit this mechanism. And this is just to point out again, there are the flames, right? That if we just take these cells, and this is the experiment in this paper, and transplant it into the infarcted area, you see some success, but the cells don't survive, right? The cells aren't surviving. They're not putting new blood vessels into the system. So we first then started to look at, as we make our materials, we can do two things. We can change all that synthetic chemistry, which I won't go into tonight, but I'd be happy to answer any questions afterwards. We can take these systems and we can change the amount of the crosslinker to make some stiffer and some weaker. We could also change the peptide chemistry. This is what we call a response surface. So we call it response surface methodology. So we can tune the stiffness and we can tune this chemistry. This is the adhesion peptide that we use. has this putative RGD sequence. What you're looking at here is the staining of the cells that represent new blood vessel formation. I have to be careful, but for this audience, I'll call it new blood vessel formation. It's really a network of these blood vessel cells, right? And these are just conditions in which we made this system. And we can get a quantitative measure of this with microscopy, not only look at the tube length, if you will, of this network, but also the thickness. And the thickness is on the range of a small capillary or a little bit larger like a capillary. So that's of interest to us that we can do this. It's important to point this part out, which is if we do the same experiment that I just described, and if we put the protein of interest outside the gel, we don't get a good response. If we put it inside the gel, remember this also has cells, we don't get a good response. It has to be coordinated and presented off that heparin. That's what happens naturally biologically, so it's not a surprise, but it is a surprise to us in some ways how poor this one behaved as well. And this is just repeated in this scenario. So what's happening in this system that makes these vascular-like networks? And for those of you that maybe don't see this kind of work all the time, this is the process. You do a lot of work in the lab, if you will, with cells, and then you move into experimental animals under, I think, very cautious terms to make sure you have something that works. So why would these things work so well? So what happens is the cells are there, and we put this growth factor in, but the cells start synthesizing all these other ones. So we can enzymatically digest the sugar, if you will, which is the backbone of the network, and collect these proteins. And then we can put them on what we call an array that captures the proteins, and we can quantitate the amount of those proteins. And that's what you see here. These are just protein names. This one's really important. This is vascular endothelial growth factor. This is a molecule that helps basically the vascular endothelial cells produce new blood vessels. These are other really important growth factors: hepatocyte growth factor, binding proteins for IGF-1, insulin growth factor. They all help with proliferation of the endothelial cells. ECs are endothelial cells, vascular stabilization. Don't worry about this. This is our coding of the different material properties. I'll show you an example of why all these different tests were important. What is important is this is the full material that I have been describing. It has the peptide, the heparin, and the TGF-beta. That's why it's PHT, and an HYA is hyaluronic acid. So when we look at our experimental approach, then we take the cells and the implant and we put them in the hind limb of a mouse. We have them genetically encoded with reporters, so they express this protein, green fluorescent protein, which means we can just examine that with microscopy. And then we also have a luciferase reporter, which basically we can non-destructively do bioluminescence and image the growth, basically, in this case, between 0 and 32 days. Red means more intense signal. That's the signal you're tracking, meaning we've done our job. We've kept the cells alive, basically, for over 30 days. If you remember, I started this part of the presentation out saying that we could nominally not really keep cells alive more than a 24 to 72 hour window without hydrogels, maybe a little bit better with hydrogels. So this was a remarkable result in our minds. And so we did, the end of this experiment is we inject a probe called isolectin, and the isolectin gets injected in the heart. And so if the blood vessels of the animal have married and hooked up with the blood vessels of that in the implant, we will see this isolectin stain in the implant, and that's what you see. You see the blood vessels now are stained with this isolectin. They're mature and thick blood vessels. This is really a nice, robust experiment. This implant is actually surrounded by blood vessels. It's really quite an interesting result, right? And so We have other important information. This is, if you go this direction, this one doesn't have the TGF-beta. The T is missing. This one doesn't have the heparin. You can see the results are no blood vessel formation under those conditions. So we need that complete system. We can also go further and just say that if we look at something like collagen production, and again, this is the isolectin perfusion stain. Collagen production, specifically collagen 4, if you remember when I was describing hydrogels that we use, I said the basement membrane, the matrix that is popular to use to culture cells and develop protocols for tissue engineering, uses a lot of collagen type 4. And you can see there's quite a bit difference between all of these conditions, with the full loaded system being the best. So in a sense, we've talked about 1, 2, 3. We haven't talked about degradation kinetics. I do want to say something briefly about that, because this it was essentially a surprise for us in the lab. As we use these cross-linkers, and don't worry about the chemistry, this is sort of the Goldilocks sort of story. This one's the slowest, this one's a little bit faster, this one's really fast. So it's fast, intermediate, and slow. And nobody had done an experiment in an animal where they compared this type of scenario, just changing that small part of the material design. And we thought, well, we should do this because we know in the literature that these are different reaction rates. These are essentially enzymatic kinetics. Those of you studying biochemistry know the Michaelis-Menten kinetics. And so these are the relative reactivity or degradation for these proteins that are produced by the cell to remodel that matrix. So we called it remodeling by cellular demand. So the cells now produce this material, these proteins. These are called matrix metalloproteinases, these MMPs. And it actually allows for the degradation of that matrix and the cell to move through while it's synthesizing molecules of interest. And we already talked about some of them, but what I really want to highlight is these actually are just too fast. And a number of colleagues use these types of cross-linkers. The one that we chose is this green one, and you can see there's quite a bit difference in this relative protein expression for all of the important growth factors for blood vessel formation. And here's just a quantitative plot of something like that. Time in days, this vascular endothelial growth factor goes way up for this condition and is not so good for either of these conditions. And then that story is borne out in vivo, where you see nice, really growth and bioluminescence. This is only the first 7 days. And you also see now the blood vessels forming in this particular experiment. Not as nice as the other one, because this one only went 7 days as opposed to 30 days. But the story is the same. And then we can do similar stains with those different cross-linkers and show that this one really has a profound effect on the type of proteins that the cells make. Both collagen type 4 and laminin are very important proteins that cells, in particular cells like vascular cells, use when they're developing new networks. The mechanism for this, I think I'm gonna bypass and just tell you that the cells have a co-receptor called endoglin, or CD105, that works with this TGF-beta, and we go down this signal pathway to activate these cells to become endothelial cells. This also happens with human cells. These are cells from patients, and we've also confirmed this with cells we create by induced pluripotent stem cell differentiation into endothelial cells. Okay, so that story is a good one in a sense that we have figured out a way to make a synthetic system that keeps stem cells alive. Where does that go next? It's going into sort of preclinical animal models where we would look at limb ischemia, so patients that have, typically diabetics, that have lower limb poor circulation problems would receive this combination of therapy, and that should start to promote angiogenesis and new blood vessel formation, as well as that whole cardiac story where we'd inject that into the heart. So looking at time. There's another type of study that we do with this system, Same sort of hydrogel, but I call this sort of the Jason Bourne experiment. This is where we can inject the hydrogel with fat cells. These fat cells are what's called beige fat. Most of us want to get rid of fat. That's white adipose tissue, or WAT. We want to enhance BAT. That's beige adipose tissue or brown adipose tissue. That's the thermoregulator fat in your body. Fat's pretty important. Everybody's like, I want to get rid of all my fat. It's like, no, you don't. You need your beige fat. It's actually, you could almost call it an organ that's essential to how your body functions. So we did the same thing. We took these cells that were beiging, we added them to our hydrogel. The chemistry is different, but the same idea takes place. We injected it into the same portion of an animal. This is basically an IR camera, so it's looking at infrared. This is the brown or beige fat cells producing a lot of heat, right? So they actually can produce heat and keep basically this animal or Jason Bourne warm if things get really cold, right? So in a cold challenge, when they're exposed to 4 degrees, when they get this transplant, they maintain basically very close to body temperature, much more so than without that system. They also have the advantage of basically, if they're fed, the animal's fed a high-fat diet. The high-fat diet doesn't increase body weight, so you actually take on less body weight, and your glucose levels can be dropped by using this transplant system. So we're funded to look at this as a way to maybe regulating diabetes and obesity. And the holdup, again, is adding cells here. So we're trying to do this just with the material that we would inject into patients, and that would enhance brown fat formation. And hence, it would help with these scenarios, not cold temperature, but basically body weight and glucose metabolism. I think the last thing I'll mention is volumetric muscle loss. This is in collaboration with George Christ at University of Virginia. Volumetric muscle loss is a big problem, especially with patients that have been in the current wars, both limb and facial, craniofacial damage occurs, and repairing muscle is challenging when it's large. So we call that volumetric muscle loss when it's large. So George creates these defects in the tibialis anterior, which is the lower limb of the rat. We take our hydrogel. It is important to note, this time we have no cells, and we didn't add any growth factors. This system is sort of the ideal from the standpoint of readiness. So the Department of Defense is funding us to have these materials dried. Remember those little spheres I showed you? They could be dried. This could be opened in basically a casualty setting, either an accident or battlefield, and it can be placed on the muscle injury. It hydrates with blood and so forth, and in this case, I'll show you an experimental animal. It regenerates the muscle quite well. So in this case, this is the injury here. This one had the hydrogel coming down here. This is the control. This is what normal muscle would look like. This is what our hydrogel therapy looks like. This is what happens when you don't have any therapy. You can see the scarring on the surface of the tissue. This looks nearly repaired. Not the same here, but certainly repaired and functional, and I'll show you that in a moment. I'll skip this in the interest of time and just tell you the muscle fiber size is enhanced in the hydrogel. The blood vessels that are surrounding the new muscle that's formed are the same as the control tissue, and you can look at it this way or in these different sections pointing out that information. But most important is the mechanical force is the same, and it's not the same, but it's getting to functional recovery. And so this is the important experiment that we talked earlier about innervation, and that's the problem with ex situ tissue engineering. So in situ tissue engineering, especially in muscle, where there's a lot of progenitor and satellite cells, we get innervation into our system. And we know that because we test the force, George Christ tests the force, by electrical stimulation of the peritoneal nerve. So we're actually stimulating the nerve, and then the foot twitches, right? And so in that case, you can see enhancement by the hydrogel versus no treatment. This level of enhancement is consistent with studies of basically functional improvement and recovery of gait correction or gait to a normal fashion in these experimental systems. So to summarize, just to say we've created a lot of these experimental hydrogels. We've for years known about these top two, maybe these red ones or something in the last 5 or 6 years we've discovered. These are really important. We've done all these things that I've explained in terms of we can keep stem cells or stem cells now differentiated into a cell type of interest, so an angiogenic cell and endothelial cell. We can keep those alive for the duration of our experiments. We can use a biomaterial basically to stimulate tissue regeneration in vivo. That was the muscle experiment. And then, we've demonstrated this in 3 different in vivo models, angiogenesis in 3 different models, and also this angiogenesis and nervation. This is this was the holy grail I was pointing out at the beginning of the presentation. I think I'm going to stop. This is about right time. If you're interested in this part, I will just tell you simply, we make these organ chips. We use what's called induced pluripotent stem cell technology. We microfabrication, which is what you use to make a computer chip. We combine the stem cells and the microfabrication to make these chips to ask all sorts of interesting questions, including those in regenerative medicine. And that looks something like this when it's all said and done. The kinds of things that we're interested in. Go back 1 slide. Things like we talked about regenerative medicine. Doing this in this kind of sort of organ technology on a chip is beneficial because we don't have to race through experimental animals. So it's more not only cost efficient, it's more ethical in that case. But we're involved in many different things: countermeasures and threats, gene editing, EPA toxins. We just wrote a grant on getting rid of these nanoparticle plastics in the ocean and toxicity into patients. We do a lot of work with drug development, and we're working with a group on organ storage and transport. All from taking everything away from experimental animals and moving it in vitro in a chip, and all of these and everything's done with human cells. So with that, I want to thank all of you for spending your Wednesday evening, early evening, I suppose. It's a pleasure to come, and I hope I stimulated some thoughts, and I'd certainly have time to take some questions. I need to acknowledge these are all of, I don't really do the work. Maybe people that are from the outside don't know that. Professors just mentor students. We have really a lot of good students. And some of these folks are now at professors at different universities already. Syracuse University and Wash University in St. Louis. Yergim Yergazarians is a collaborator at UCSF. George Christ at University of Virginia. And Andreas Stahl is my collaborator at Berkeley. We have funding from all these federal agencies, which makes the work go forward. This is really expensive work because stem cells are expensive, and handling them with reagents is also expensive. So with that, I thank you. Thanks for Robin also for the invitation and keeping me on time, which I wasn't with traffic, but I apologize. But I'll be happy to stay afterwards to meet with the students that I couldn't meet with earlier. Thank you.
And actually, we do have a few moments to take a few questions from the audience. So if there's anyone who has a question they'd like to present to Dr. Healy, I'd be happy to bring the mic around. All right, well, it looks like no questions from the audience. Dr. Healy said he'd be welcome to stick around for a couple minutes if you have questions for him personally. With that, please join me in thanking Dr. Healy once more for his time this evening.